All right, ladies and gentlemen, welcome back. So today we're going to talk about just the basics of viral replication. So first off, let's just talk about the fact that viruses are what we classify or talk about as obligate intracellular parasites. So let's sort of break that terminology apart for a second. They're parasites, which means that they are ultimately going to require a host and will ultimately damage their host in benefit of themselves. They're intracellular parasites. So if you know that prefix intra, that means within. So they have to be within a cell in order to complete their life cycle. And then they're obligates. They're obligated to be within that cell in order to do their replication, to complete their life cycle. So they can't be anywhere else. This isn't an option for them. Um, so that means that they can only replicate within their host cell. We keep them out of that host cell, we prevent them from replicating, we prevent them from infecting. So each virus has a very specific host range. They can only attack a certain limited number of host cells. Those cells oftentimes are limited to either a particular species or a particular um, marker on the cell membrane, which is oftentimes limited to a particular species or a handful of closely related species. Um, it's why more often than not you can kind of be sick on the couch at home and not have to worry about passing it on to your pets. There are viruses that can transmit from human to animal or vice versa, but generally speaking, most viruses um, stick to a particular type of host. So when we talk about viral replication, we can talk about some general features of replicative cycles that are pretty much seen across the board. And then we'll sort of dive in as we talk more today about more specific types of replication. But generally speaking, all viruses have to get their viral genome into their host's cell. Once that genetic material enters that host cell, that genetic material then contains the information necessary to make more of its genetic material while also manufacturing additional viral proteins to build that capsid to then go make more viruses. When that virus infects a cell, it's also going to take over all of that host cell's machinery. Their enzymes, their ribosomes, their tRNAs, amino acids, ATP, pretty much everything that cell would have been using for its own life-sustaining processes, the viruses are going to come in, either destroy them to get them out of the way, or more often just commandeer them and take them on to do things for themselves, to do things to make more viruses as opposed to continuing that that life cycle of that cell. Those viral nucleic acid molecules and those capsomeres then spontaneously self-assemble within the cell once they are built to then make new viruses. So when we look at the basic pieces of any virus's life cycle, it's infect its host, take over its host, build more of itself, release more of itself to go infect other cells. That's pretty much the crux of how these work. Now we can get into more detail about specifically what certain types of viruses do, which is where we're going next. So remember I mentioned in the last video that we understand bacteriophage life cycles probably the best, and so we use those as models to understand other viruses. So we're going to start with what we know well, and that is those life cycles of bacteriophages. So interesting thing with bacteriophages is that they can fall into one of two cycles. Either they fall into this lytic cycle, which is what we'll talk about here first, or they are going to fall into the lysogenic cycle, which we'll talk about in a moment. So when we look at phages that go through the lytic cycle and the lytic cycle only. They're going to replicate and ultimately cause death to their host. This really is the basics of viral replication. Attach to my host's cell, right? Inject my genetic material into my host, degrade my host's DNA so that it can no longer be utilized to control the function of that cell, take over the host cell's machinery, enzymes, ribosomes, amino acids, the whole 9 yards, build more viral DNA, build more protein capsules, assemble more viruses, and as that cell gets so engulfed and so, the word I'm looking for, so filled up with those new viruses that are being assembled, they will eventually lyse or break the cell. So once that cell wall busts open, then those bacteriophages are free to now go infect more cells. So we talk about viruses that, or bacteriophages, I should say, that only participate in the lytic cycle as virulent phages. They just attach, inject, take over, build new, release. That's it. That's all they do. Now, bacteria actually, interestingly enough, have evolved defense mechanisms. They don't have an immune system like we do, but they have evolved ways to try and defend themselves against these viruses. And what they have evolved are things called restriction enzymes. And tuck that word away for yourself for later this year if it's not something you're already familiar with, because it's going to come back. Restriction enzymes are enzymes that these bacteria cells have evolved over time to specifically cut apart viral genetic material at specific sequences. So certain phages that they will get infected with regularly have a certain set of genetic information, have a certain sequence of DNA or RNA, and those restriction enzymes have evolved to cut the DNA apart of that virus. And if they can do that, then they stop the process right here. That genetic material comes in, those restriction enzymes chop apart that viral DNA. It's no longer accessible information, and so the cell has prevented itself from being infected. Why do I say hang on to that term for later this year? When we get to talking about biotechnology, restriction enzymes are going to come back, because we're going to talk about how humans actually use those restriction enzymes now to be able to create genetically modified organisms, plasmid rings. We use it for a whole host of technology that humans now utilize for themselves. So we're going to come back to that term and talk about how that works later this year. If it's not a virulent phage, then perhaps it ends up in this lysogenic cycle first. And this is a pretty clever cycle, because what it ends up doing is we still see this attachment, we still see the injection piece, but we're not going to break down that host's DNA right away. Instead, that viral DNA is going to come in, and we're going to talk about it as a prophage. That prophage genetic material incorporates itself into the chromosome of that bacteria that it's infecting. So here's that prophage. And what that bacteria does, nothing really here at this point. It doesn't even really recognize that that genetic material is sitting there. But what it will do is go through its normal life cycle, and it will reproduce. And as it reproduces, that means it needs to make copies of its genetic material. And so as it's copying its own DNA, it's making copies of that prophage. And that can happen 10, 20, 30, 40, 50, 100 times before that prophage DNA does anything. And so now I've been able to infect one cell and ultimately may end up with hundreds of cells that are carrying around that viral genetic information. Eventually, something happens in that bacterial cell's environment that forces that prophage back out of that host chromosome, and we induce that lytic cycle, and so it'll pop back over here. So we'll sort of see this attachment injection will pop off into that lysogenic cycle. Something will force that bacteria back into the lytic cycle, and then we complete this. So they'll break up the host genetic information, take over the cell, synthesize a bunch of viruses, and release them. But the big difference is that lytic cycle, it was one virus destroying one cell. When we look at lysogenic pathways, we're looking at one virus infecting an initial cell that replicates potentially into hundreds of cells before those hundreds of cells now end up in that lytic cycle and get destroyed. So it's in a lot of ways more efficient if the capacity is there, and I'm going to use some terminology here that's not truly biologically correct, but that those viruses are sort of patient, if you will. Um, Again, they don't have a brain, they don't have cognitive thought, but you can kind of think of it that way, where they're have evolved to sort of do this sneak attack. They infiltrate those cells, they replicate themselves without causing any initial damage, and then they just blow up that colony of bacteria from the inside. So we talk about these guys as temperate phages. So lysogenic viruses are temperate phages as opposed to the lytic cycle only viruses being virulent phages. So again, it's just really kind of clever the way that this has evolved. It takes a little bit more time for replication to occur, but the evolutionary benefit is that I can make a lot more of myself, um, starting off with a lot less, um, when they go through this lysogenic cycle, as opposed to strictly sticking into that lytic cycle option. So when we kind of compare them, right, virulent or temperate phages can go through that lytic cycle, but when we talk lysogenic cycle, it's temperate only. So temperate viruses will go through that lysogenic cycle and then pop over to the lytic cycle when some sort of environmental trigger forces them to do so. But virulent phages are stuck in that lytic cycle and that lytic cycle only. Lytic cycle, it's attach, destroy, reproduce, bust out. All right. Lysogenic, though, we're going to incorporate that, go back that viral DNA into the bacteria chromosome, replicate right along with those daughter cells, and then eventually get induced to leave that chromosome and initiate that lytic cycle.
In a lysogenic cycle, viral nucleic acid becomes part of the host cell's chromosome and is replicated with it. Eventually, the virus enters a lytic cycle and kills the host cells. A bacteriophage attaches to a bacterial host cell by locking onto a specific receptor site on the surface of the host cell. The virus then injects its DNA into the host cell. Once inside the cell, the viral DNA inserts itself into a specific site in the chromosome of the host cell. Viral DNA is then called a provirus. During cell reproduction, the host cell copies the provirus genes along with its own DNA. The provirus is inactive at this time. When the cell divides, both the host genes and the provirus genes are passed on to the two daughter cells. At any time, a provirus may leave the host chromosome and enter a lytic cycle. When this happens, the host cell's DNA is broken down and new virus parts are produced and assembled. Eventually, the cell ruptures and new virus particles are released.